same thing?
Okay. First one says we need to be able to explain the basic steps of genetic engineering. So first of all, what is genetic engineering?
The process of isolating the gene from the DNA of one organism from the DNA of another.
Right, so we're going to take a gene out of one thing and stick it into another.
Yeah.
Now, the basic steps there, number one, you got to isolate the gene of interest, the thing that you want to take out of the organism, the traits that you want to pass on to the other organism. So, for example, if we're talking about insulin, putting that in a bacteria, we have to isolate the gene in humans that makes insulin. We have to cut that out.
Is that step 2 or is that
Step 2.
Can we put this in a...
Yeah, should be. Okay, and then we have to take the host organism, the one that's going to get the gene, we have to cut their DNA in a way that we can put that new gene in. So we talked about the restriction enzymes and the sticky ends. We'll do that in a little bit. But you basically have to cut a space for the gene into the other organism's DNA. And then you have to get that gene in there. So we have to use a vehicle or a vector to be able to bring that gene into the cell and put it into the DNA. You guys learned a little bit more about vectors when you were doing your gene therapy assignment yesterday.
We have to know how much stuff.
Just that you don't need to know any like the specific types or anything. It was just that vectors are things that can be used to put DNA into a cell. Okay, number 2, explain the use of restriction enzymes and the pattern of cuts they make to DNA. So the whole point of restriction enzymes is they always cut where there is a specific sequence. They'll cut anywhere that sequence shows up. Some restriction enzymes are going to cut just straight down through the DNA. But remember, the useful ones are the ones that make those jogged cuts, sticky ends. Because if you cut one thing with sticky ends, and you cut another thing with the same restriction enzyme, so it has the same sticky ends, and it's a lot easier to put those things together with ligase enzymes to seal them up. So genetic engineering is one place where the use of restriction enzymes is important, because we have to use them to both cut the DNA, the gene out that we want, and also cut the DNA to have a place to put the gene back in. What's another place that we use restriction enzymes? Another instance that we talked about. Anybody? Yeah, the DNA fingerprinting. Because remember there, to be able to run the gel and get those DNA to separate out based on their sizes, we first have to cut that DNA up. So, down here we'll talk about the steps of DNA fingerprinting, but that's the important step, is using the restriction enzymes there. So, both in the genetic engineering and putting a gene into a new organism, but also in DNA fingerprinting to cut up those pieces of DNA so they'll move through the gel. What do we call an organism that has DNA from another source? Transgenic. Okay, and remember we call their DNA recombinant DNA because we basically put it back together. So transgenic organisms have recombinant DNA. Okay, This probably actually makes more sense if we talk about 4 first, so we'll come back to 3. But let's say we find some blood at a crime scene. What are we going to do to make a DNA fingerprint from that blood?
We have to multiply it.
Okay,
What?
so collect it is kind of the obvious first step, and that might be too obvious. We're going to collect a DNA source.
Put it in a little thing.
Hold on, man. But yeah, then we multiply it. And how do we multiply it? How do we multiply it? Polymerase chain reaction, or PCR, right? Remember, PCR, basically, you just throw a whole bunch of extra bases in, and you throw in some DNA polymerase enzymes, and they're going to read all those pieces of DNA, and they're going to just make lots of copies of it. That's the way polymerase chain reaction is. The basics of it. So we collect, then we multiply using PCR. Once we have lots of DNA, then what do we do?
Put it in
Not yet. We got to cut it. So remember, we cut it up using restriction enzymes. Because the whole point of the gel electrophoresis is it's going to separate all those little pieces based on their size. So if you haven't cut it up, you're going to get one band. That's all it's going to be. Okay, so step 4, that's when we electrophoresis it. And the other thing that we should know here to make a good DNA fingerprint where we have lots of things that we compare is that we're going to repeat with a new restriction enzyme. So we'll repeat with a different enzyme. We'll compare all of those different panels so that we get a good basis on what the DNA is actually, what bands.
How many times do you repeat it?
I don't know what the standard is. Oops.
Okay.
Yeah. 5 or 6 different restriction enzymes. I would say at a minimum. Okay. Now let's talk a little bit more about the gel electrophoresis. So remember, the point here is that the gel has pores for those little pieces of DNA to move through. And the smaller the piece of DNA, the easier it is able to move through the pores, the quicker it's going to move towards the other end. Okay, so the gel has pores, and the DNA pieces move through them. Why? What makes the DNA pieces move?
Isn't there cut off with the negative charge?
Yeah, electricity, basically. We have a difference in charge. Like a lot of things in chemistry, a difference in charge is going to make stuff move from one place to another. It's kind of related to magnetism. But basically here, electrical charge is what makes the move. Okay, and I would absolutely, positively, definitely know that the bands that move the farthest are the ones with the smallest pieces. Bands that move The smallest are the ones with the biggest pieces. Okay, any questions there? All right, number 5. If I give you a DNA profile, you should be able to match that up to, if there's a crime scene with some suspects, you should be able to look and see, okay, this is the person that the DNA matches to. But you should be able to look at the other ones and go, this is why these people rule them out, that they were not the suspect. Well, they may be suspect, but they were not the person.
I remember like that, where those guys with babies, whatever. So it's like the mom, dad.
Paternity test? Okay, we'll talk about that in a minute. Okay, but for the basic, where you're using a DNA profile to match it up, remember you're going to look for, if you're matching it up against one, we want to see all of the bands match up. We don't want to see any bands that don't match up. So you're both going to be looking for gaps that occur in the other person's DNA where there is a band in the sample, but you'll also be looking for bands that occur in the other person's DNA that are not in the sample. So those are the things we're going to go, well, he's got a band, there's no band in the sample, he's got DNA that the sample doesn't have, that doesn't work. Okay. Now, when it comes down to paternity, though, that's a slightly different. Because there, we're not looking for a perfect match of the DNA profiles. Why? Right, half of the DNA comes from the mom and half comes from the dad. So when we're looking at a DNA profile for a child and trying to figure out paternity, remember, we have to have the kid's DNA, but we also have to have the mother's DNA so we can look for the bands that the mother does not have, because those are the ones that we actually want to match up and try to the potential fathers and try to use to confirm who the father is. Because those bands that the kid has that the mother doesn't have, right, they have to come from somewhere, so they must have come from the father. So those are the ones we're going to want to look at. But to do a paternity test, you have to have the mother's DNA, you have to have the kid's DNA, you have to have the father's DNA. That should seem obvious, but I know that at certain places where they do paternity tests, a lot of times they'll show up without the kid. They're like, well, we need their DNA too.
Oh my God.
Okay? Alright, now yesterday you guys did some stuff with gene therapy. Now, you went much more in depth than you're going to get to know for the test. You need to know the basic ideas of gene therapy. So, what are the basic ideas of gene therapy?
They just take something like a good or get and replace the way the
Okay, that's the basics of it. The basics of it is, hey, we've got this gene that causes this debilitating thing like cystic fibrosis. Let's kick that gene out and put a good gene in place so that people don't have cystic fibrosis. That's the whole
Would your
point.
kid be born with the gene that they put in then, or would they be born
So if you had, say you had cystic fibrosis and you got gene therapy, it would depend on how ubiquitous that gene replacement was. Because they're going to focus on the lungs, because that's where the cystic fibrosis are. To be able to pass it on to your children, of course, you would have to have that replacement in your reproductive organs as well. Females, probably not as easy, because as we talked about before, you're born with all the eggs that you make already in that state, whereas males,
so your
because they make
kids
sperm.
can still have it even if you got
Right. Males, because they're making it continuously, if you replace the genes in those stem cells that create the sperm, then impossible. Okay? And other than that, it's basically the same idea as making a transgenic organism, right? Have to have a gene that we're going to put in there. We got to cut it out, put it into the DNA. But the biggest thing is, especially for gene therapy, it's a little bit harder to use a vector. Because unlike having just a single bacteria cell, where we can use, we can maybe just even stick a needle in there and squirt the DNA in, that's not going to work when we're talking about replacing the DNA in all your lung cells. So vectors are going to be things that are used to basically put the DNA, or let's say put genes, into cells. You guys looked at some vectors yesterday. What kind of things were they talking about using as vectors?
Herpes.
Different types of viruses.
Lysosome.
What's that? So a lysosome, yeah, different type of a molecule, but it's not a living molecule. What would be a drawback or a negative for a con for using a virus as a vector?
Yeah, putting a
Well,
virus
that's
like
like
every
specific.
What's that? Some of them aren't specific, that's true.
You have to keep getting injections. Most of them are just saying.
Okay. Yeah, depending on what type of virus it was, whether it was a retrovirus or not. You may have to get What's that? Right. The body is going to think that you're putting a virus in there. Now, are you actually putting a virus in? No, because they've taken the DNA that makes the virus F. But yeah, the problem is your body's not necessarily going to know the difference. And so your body's going to go, hey, there's this, this foreign invader. We're going to kill that thing off, even though what we're trying to do is help it. So that would be a major drawback for using viruses as vectors is the body's immune system. Trying to kill the thing that you're trying to use to change the genes. Okay. And those are the basic things that you need to know about gene therapy. So if we're putting genes in new cells, then you got to use vectors to do that. Okay, and then the last one says explain the basic process of cloning.
Start with one cell.
Okay. One cell from what? Whatever you're cloning. So let's say we want to clone Jason.
Let's repeat
Let's say we want to clone Jake.
that.
So the first thing we need to do is we need to get a cell from Jake. Does it matter what kind of cell we get? Really. You do have to be a little bit careful. A red blood cell would not work because red blood cells actually don't have a nucleus in them. So there's no DNA.
What if you just got blood?
You could find cells, right, white blood cells and other cells that are in the blood that you could use, that's true, but a specific red blood cell would be a great choice. Which is too bad because red blood cells are fairly good size. But, yeah, we can take a skin cell from him, we can take a muscle cell, basically anything that's got DNA in it.
Could you take two cells? What would happen?
Yeah, you don't want to put more than one chromosome of each chromosome in a single cell. I mean, you can do multiple new cells. Yeah, we're going to do it lots of times. We're not going to go, once, that's good enough, because a lot of times they fail. We're going to try it for multiple days. So we're going to take some cells of his, we're going to pull out the nucleus of his cell. And where are we going to put it?
In the cell.
Into an egg cell.
Into an ova. So we need a female to donate some ovum, some egg cells, to be able to be used. But before we can stick that DNA or that nucleus in, what do we have to do to the ovum? We have to take out its nucleus. So basically, we're going to take the egg cell, we're going to swap out its nucleus for the nucleus for Jake's cell. And then what?
It grow.
Let it grow. We got to do one thing before we let it grow. Everybody remember?
Water.
Not water. Not water.
It's on the plant.
No. Everybody remember?
Oh yeah, have to put it.
We got to shock it. So you use an electrical stimulus to kind of make the cell decide that it needs to start growing. What I think is happening there is basically causing some plants to start splitting. But because this is a ovum, and it now has a complete nucleus inside of it, when it starts splitting, it starts differentiating, and it grows up. As long as you put it where? In a womb. So, if you put it in a petri dish, what are they going to turn into? The cells, right? Stem cells. So the major, not more. The major difference, really the only difference between creating a clone of Jake and creating some stem cells that we can use is where we stick them after we've shocked them and made them start dividing. Putting them in a womb that they can grow, nourish, and become a human. We have a different outcome than if we just stick them in a petri dish and let them divide using the stem cells. That's why those types of procedures are not legal in the United States right now, because we're still trying to figure out the morality of that whole thing. We got these cells that if you stick in one place, they will become human, different than any of the rest of you. If we stick them in another place, then they're not. That's why there's a moral issue.
So we can make clones of that.
Well, the other moral issue is that, like I said, there's a lot of ones that don't turn out.
Did you clone your people in the womb still? Yeah, yeah.
Well,
That would be like
it's always going to be a little bit behind. So I
a
don't.
little bit, not that much. Like if we just have it born on the same day, like it'll just be a couple weeks.
I don't think that would necessarily work out terribly well. But yeah, I mean, anytime you can get a cell of DNA. Okay? Oh. And then remember, don't forget, real clones, if we're going to really clone a person, you still have to grow them up. So you won't have a clone of Jacob for quite a while. The other thing is, remember, it's not necessarily going to be like Jacob. You'll look like him, you'll have the same DNA. You can maybe frame him for a murder as long as they don't find it for another however many years. But there's no telling what he's going to be like personality-wise. So maybe we should clone Jason, see if we can get it. Check him out right this time.
Yeah, that's so.
Okay, any questions there?
Nope.
Yeah, we went back and talked about that, right? Yes. Okay, well, good luck studying for your test. Just a minute. Tomorrow we have first hour CRT, correct? So I imagine I will see some of you. But if you don't come in, at least take some of that time and actually study for your test. And yes, there is a question of the day.